Good afternoon, everyone. Thank you so much for joining us for our very first Lunch and Learn series sponsored by the CART Fund. So at this point, I want to introduce Dr. Karl Herrup and allow him to take over and make his presentation.
Thank you very much. It's a wonderful privilege to be here. I think it's a great effort, and I'm really pleased and grateful that the CART Foundation is doing this sort of outreach, not just to its own members, but to the broader community. But let me start first of all by saying that given that this is the first presentation, I thought I would take a broader view of the whole area of Alzheimer's disease and Alzheimer's disease research. I'll actually touch very little on my own personal research, but walk you through some of the more, I guess, policy-oriented opinions that I've had over the years, and let's see where that takes us. So you can see from the very beginning, I've actually renamed the festivities as lunch a la carte because I just like that a lot. So you'll pardon my pun. But let me start as I threatened I would with some personal background. As Tiffany said, I have a very peripatetic professional career. My prehistory, which history starts for me in 1992, as that was arguably the year I became an Alzheimer researcher. Before that, I was actually a developmental neurobiologist. I'd studied the earliest moments of brain development. And I still think that early history informs the science I do today. I did spend a number of great years in Cleveland, which is where, and in the Alzheimer's Research Lab, which is where I really cut my teeth in and dug into the research surrounding Alzheimer's. And of course, it was the place where I got my CART grant. But what was fascinating to me and what really formed my thinking as a researcher is it was my first introduction to the clinical disease we know as Alzheimer's. I am by trade a geneticist and a cell biologist. So it was wonderful for me to be able to participate with our Pierre-Luigi Gambetti, who was our neuropathologist at the time, and attended nearly 100 different brain cuttings. So you really begin to understand what this disease does to the brain and make that transition between what I do as a cell biologist and what actually happens to people. But it was more than that in Cleveland because the center prided itself in being a broad-based center. And it was one of the first in the nation to have a focus on research into the problems and processes that the caregivers of individuals with Alzheimer's disease have to go through. As Tiffany mentioned, during that time, I became despite my origins in early embryogenesis, I became director of the Alzheimer's Disease Center for 6 or 7 years. And it was that CART award that sharpened my interest in the brain inflammation, which to this day remains an important part of the what goes on in Alzheimer's disease. I moved from there to Rutgers. It was as much because I was given an opportunity to lead a group of cell biologists and neuroscientists. But commensurate with that, I actually left a medical school for the first time in my professional career. I'd always been in a medical school. And with this move, it really freed me to focus totally on the basic science. But I never lost the connection to clinical science. But the focus on basic science, I think, really helped me quite a bit. I'm not going to talk about it at all today, but I will point out that I met a landscape architect who specializes in the design of Japanese gardens. And I have to this day whoops, let's see if I can get this back. An interest in their impact on the symptoms of Alzheimer's disease. And then just quickly to summarize, the move to Hong Kong, I felt it gave me a really strong international perspective on how Alzheimer's disease is not just an American problem. It is a problem that faces literally the entire world. The world sees it differently, but it does not avoid the scourge of this disease. And in 2019, I returned to Pittsburgh. And of course, then COVID hit, so I don't have much to tell you about my time here. But that's my background. And what I'd like to do next is ask a very simple question to you. You don't unmute yourself, but just sort of think about this and prepare maybe an answer for when we do the question and answer session. But if I asked you, what is Alzheimer's disease, what would you answer? What would you tell me? In writing my book, I asked that question of many, many people. And what I can assure you is that just about everybody gives me a different answer. And I think tellingly, that's especially true for my scientific colleagues who really are almost all over the map when it comes to giving me a concise definition of what Alzheimer's disease is. But I maintain that the really relevant answers come from people just like yourself. You know, what is it that you think of when you think of this label of Alzheimer's disease? You know, what do you focus on? So classically, one would focus on the loss of memory, I know, but other features of the disease impact you and impact the person with the disease, as I've listed here, and there are many more: the loss of the ability to live independently, the behavioral changes that are not just that are actual symptoms of the disease, and the loss really of personhood. An individual who you've known, perhaps, you know, undoubtedly all your life, almost feels like they're slipping through your fingers and disappearing. But my bet is, and I put $100 down, I'd bet $1,000, I'm sufficiently confident, that your answer would focus almost entirely on what I would call the symptoms of the disease, the manifestations of the disease in a living person. And what I want to focus on, and I'm going to take you down a dark path but I promise to bring you back by the end. If you ask a scientist that same question, I find that the answers by and large end up being divorced from this lived experience. And I'm going to go through the explanation of why I think that is and how it really gives us insight into how we should chart our path forward. So here's, you know, obviously I could spend an entire graduate course in the topic of Alzheimer's disease, but I've tried to simplify it into one slide. So you'll bear with me if I oversimplify it. But can everyone see my pointer when I go like this? Great. All right. So this is the man after whom the disease is named, Alois Alzheimer. And this is his signature case, August Dieter. And what Alzheimer noticed was that August had a really aggressive and virulent form of the disease and died rather shortly after being diagnosed. What Alzheimer's was able to do was then take her brain and stain it with silver stains. And what he saw when he looked there were these unusual deposits, what he claimed were unusual deposits of these plaque-like things, and the ghosts of old neurons that he called tangles. We're going to ignore the tangles for now, but the plaques ended up becoming a central feature of the disease. And so in the 1980s, we were able to use advanced biochemical techniques to extract the plaques and analyze them chemically, and discovered that they were actually made up primarily of a small protein, about 40 amino acids in length, which has become known as the amyloid beta protein, or A beta. And in a very exciting decade or two, scientists were able to both identify the gene that made this peptide and discover that the peptide was part of a much larger protein that was stuck in the membrane called amyloid precursor protein, and that you got A beta by an enzyme that cuts proteins right here called the beta secretase. That's not important. And then another protein that cuts right here called the gamma secretase. And it was those two cuts that freed this. And this was our scientific view of Alzheimer's disease. And what the excitement of that era led very quickly to what has become what I call the central dogma of Alzheimer's disease, which is known as the amyloid cascade hypothesis. And I'll describe it to you in the next slide, but I want to emphasize that it began life, and to this day remains, in my estimation, a hypothesis, which is that it's an attempt to explain a disease and ripe to be tested for its validity. What I say here and what is true is that it's a perfectly fine hypothesis. In fact, in the era of the early 90s, when it was proposed, I would say it was a brilliant hypothesis. But like all good hypotheses, you put it to the test and, throw rocks at it and see if you can break it. And I'm going to go through some quickly, but some evidence that basically the tests of its validity have all failed. And it is those failures that bring us to where we are today. So let me then use this same image to walk you through the hypothesis, which basically states that the A beta peptide, that little blue cylinder down here, aggregates many, many times over, and thousands of little peptides aggregate to form the plaques. The plaques in turn then produce the tangles of these dying neurons. Those dying neurons are responsible for the shrinkage of the brain that you see on brain imaging. And it's that brain shrinkage and loss of brain substance that ultimately leads to the clinical symptoms that August Dieter had demonstrated. So fast forward to the modern times, some brilliant radiologists and chemists have been able to devise ligands that can be visualized in a PET scan. And what that's enabled us to do is to actually, albeit at somewhat lower resolution, but we can actually see these deposits now in a living individual. And so if you use one of these ligands, either for tau or for beta amyloid, A beta, you can see that in a normal adult, normal older adult, there's a few deposits. They're lighter areas are the ones where the deposits are, both for tau and for amyloid. But when you get to an individual, a person suffering with Alzheimer's disease, those deposits increase dramatically, both for tau and for amyloid. And so it's strong correlation that with the advance of Alzheimer's disease, these deposits increase. It validates the approach that Alois Alzheimer suggested back in the early 1900s. But let's go ahead and let's test the hypothesis. So how would you test the amyloid cascade hypothesis as I laid it out to you? Well, one of the things you could do is you would predict it if the A beta peptide was causing the plaques were causing the tangles were causing the degeneration was causing dementia. If you stopped the plaques or got rid of them, you should get rid of the disease. But let's start by testing the proposition that if that hypothesis is true, if we add amyloid to somebody's brain, we should actually create Alzheimer's disease. I mean, that's a direct prediction of the hypothesis. And of course, that experiment's unethical to do in people. and I'm going to skip the mouse data, the experimental data for now, but it turns out that that experiment has been done by nature inadvertently. And because in point of fact, when people started PET imaging a cohort of normal individuals or individuals without cognitive impairment, it turns out that about a third of healthy seniors have plaques in their brain that would be consistent with a diagnosis of Alzheimer's. Now, that's not what the hypothesis would predict, because the hypothesis would predict if you've got those plaques, you should have Alzheimer's disease. And so if we look at it carefully, it turns out that these people with lots of plaques are at modestly increased risk, but the risk is very modest, and the time that it takes to go from those plaque positive brains to actual dementia is quite long. And if I could just emphasize that again, I showed you this slide showing the progression from low deposits in normal cognitively intact individuals to Alzheimer's disease patients with high levels of deposit. But you see this white box in the middle? Well, you see, that's there because I blotted out the central part, which is, this is an example of a cognitively normal older adult, cognitively indistinguishable from this person. And you can see the levels of deposits of both amyloid and tau are extraordinary. So this is not consistent with the amyloid hypothesis. It in fact would tend to contradict it. But let's keep going, because the hypothesis also predicts that, and this would be therapeutically useful, if we take amyloid out of somebody's brain, that we should either prevent Alzheimer's if they don't have it, or Alzheimer's disease, once it's established, should slow or hopefully even stop. And unfortunately, the finding is, after billions of dollars invested in clinical trials, we find that people whose amyloid has been removed from their brain fare no better than people who have been given a placebo. And we've tested this idea every which way to Sunday, because we can vaccinate, and that's another whole story that I'm not going to get into. We can vaccinate people against amyloid, and we can remove the plaques from their brain. We can block the secretases, which are those enzymes that free the little A beta peptide, and the plaques will go away. But no matter how we get rid of amyloid, it does not help. And in many effects, the side effects are worse than the actual placebo. So this is a very busy slide. Don't look at it. Just look here at the green. These are all the different clinical trials that have been tried, various different ways of treating amyloid. You can see in all the ones in green, these are successful reduction of amyloid. And all the ones in green, all the ones in green here, these are all the places where it made the patient better. All the blank ones are where nothing happened, and all the red ones are when people actually got worse. So rather than being a panacea or a cure for the disease, it almost looks like removing amyloid, no matter how you do it, makes people worse. So I'm going to maintain, I do maintain, that all of this data, and there's much more out there that I'm not going into, all of this data suggests that we need a reset in our field that allows us to take a step back, lick our wounds, massage our egos. We put out a hypothesis that was perfectly valid as a hypothesis, but maybe turned out not to be true. I'm going to go even further and say that we actually need to think about the very question that I asked you in the beginning, which is, what is Alzheimer's disease? And I think that has suffered as people have tried to equate it with the deposition of plaques, with those deposits that I showed you in the brain. And over the years, I have identified three what I call inflations of our definition of Alzheimer's, and I think we need to work backwards against those inflations. I don't need to go over them in great detail, but the last one I think is the most serious because in the late, actually about maybe 5 or 6 years ago, and a very august group put out an opinion piece that said Alzheimer's disease is actually defined by the plaques, not by the symptoms. In other words, if you have plaques in your brain, even if you're cognitively normal and a chess champion, you've got Alzheimer's disease. You just aren't showing the symptoms yet. And I think that needs to be rethought. In fact, it's my contention that we should be defining Alzheimer's disease by what matters to the people on this call. And your loved ones. We need to go back to defining it by the symptoms of the disease, the cognitive decline, the loss of executive processing, the behavioral changes, all the things that matter to the person in life and to their loved ones. These are what define Alzheimer's disease for us. As a scientist, of course, I'm looking for the biological basis of the disease because that's where pharmacological and other types of interventions and cures are going to come from. So from that standpoint, these plaques and tangles are clues, and they're very possibly going to be helpful in untangling the disease process. We should absolutely not ignore them, and I'm in no way proposing that we do, but we cannot use them to define the disease. And that's where I think we've gone astray, and we need to rethink and reset. And what will follow you almost naturally from that idea is that we need to reset our efforts as well. I have been in the field long enough to recognize how profoundly the study of amyloid deposits has just sucked all the oxygen out of the research room. And it isn't so much that, again, that research is bad, it's good research, but other worthwhile avenues have been actively suppressed because, as an advisor to our lab in Cleveland once told us, explicitly, if you're not studying amyloid, you're not studying Alzheimer's. And to that I say, phooey, not true. And the alternatives are, there's a long list of alternatives, candidates to be explored to help us attack the disease and tackle it where it lives. There's the role of inflammation, which is where I got started with CART all those years ago. There's the role of DNA damage and genomic integrity that interacts directly with aging, which is one of the greatest risk factors for Alzheimer's disease. The impact of diabetes, the involvement of metabolic principles and mitochondria, the wrapping of the axons known as myelin, and the list goes on and on. All of these are part and parcel of what the disease is about. And I think all of them need to be on our menu of options. And here's where I think we can bring it back to a period of optimism. So I told you I was taking you to a dark place. I think you now understand what I mean by that. But let me bring you back, because I think there are tremendous signs of progress that are emerging in the field. I think even the most diehard amyloid partisans have begun to appreciate that we need to diversify our portfolio of clinical trials. And so it is expanding these days. And I find all of that to be exciting and good. And it's not just trials in the clinic that have expanded, but our research efforts have expanded as well. And the derision that used to meet a grant that proposed to focus on something not related to amyloid has completely gone away. And funding in these, what I call non-traditional areas, is expanding, and in fact is expanding very rapidly. And then here, just to bring it on home completely, this is where the role of CART has been so fundamental in the field. I mean, it started probably as a small group out of a Rotary club, and under the brilliant foresight of Roger Ackerman many years ago. But it has placed itself really in the forefront of this reset. Over the years, the grants have been chosen for their funding and their potential for a real impact on Alzheimer's disease. And I would say this is an extraordinarily realistic and foresighted, because it's been going on for so long. And again, I'll hold up my CART grant, as Tiffany mentioned in the very beginning, as an example. It was not on amyloid. It was on the role of inflammation in Alzheimer's disease, a theory which has now gained considerable traction. And now that I'm on the CART review board, I can actually be part of the group that guides CART in a way that continues this tradition. We do still fund amyloid grants, we fund tau grants, but we also fund grants on sleep, on a host of different approaches. And I think the CART can truly be proud of the role that it's played in the field and continues to play. With that, let me take a pause and stop for questions. And thank you for your attention.
My wife was 50 when diagnosed, and they told me that possibly the disease started 15 years earlier, so that would have meant that she was 35. But you were saying, what is Alzheimer's disease? To me, I look at it as a caregiver, and I was so amazed at the ignorance of the general practitioner doctors as far as what to do for Alzheimer's. They don't know. And Medicare would make you go to a psychiatrist, which couldn't help your wife. And you had to go see them every 6 months. Most of the time, the psychiatrist was helping me because I, as a caregiver, I had diabetes, high blood pressure, a lot of things. So, but the biggest thing I wanted to ask you, doctors, when the 3 doctors came in to Columbia, South Carolina this year, one of them was talking about brain and a petri dish, and that one sentence has amazed me. I've looked it up. I've tried to read as much about it as I could. Is there a possibility, to me, that the doctor was saying that we have been doing everything on mice, and human beings are not mice. We're close, but we're not. But do we need to reset that, how we're going to do the testing, as well as what we're looking at?
Yeah, that's actually an excellent question, and I'm going to answer it by talking out of both sides of my mouth. Your fundamental point is exactly true. People are not mice. You can't actually publish that finding. It's been published already. But all kidding aside, those differences are real, and they mean that it's not enough to cure a mouse. You have to cure people. And the other side of my mouth that I want to talk out of is that if we are going to advance, we must engage we have to lift the hood and look at the pieces and parts of what's going on. And we just can't ethically do that as humans. I don't think any of us would want to. And so we have to devise experimental systems that are useful, maybe not to tell us about Alzheimer's, but to tell us about what I call the pieces and parts. So we can study cell death. We can study memory formation. We can study inflammation. The basic components are going to be the same, and so studying them in a in an experimental animal as opposed, or a brain in a dish, as your doctor suggested, is not only useful, but actually is almost critical to a comprehensive approach to a disease. So no, it's not enough to cure a mouse. Actually, we've cured mouse Alzheimer's hundreds of times. We have to translate those therapies into humans.
What is Alzheimer's disease? And I agree with you, because I'm glad you said that. Inflammation. I think that's the number one issue that we're not facing with patients. And we know that that's the cause of most diseases, right? That's the beginning of most diseases, is all that inflammation. And most people have inflammation out the wazoo, but simply because of the way of their lifestyle and what they eat. Okay.
Yeah. So it's a complicated area, but you're right. Inflammation is not just a factor in Alzheimer's disease, but in many diseases of aging. Our immune system ages just like the rest of us, and it starts to go a little haywire on us. And what was useful in our youth becomes detrimental in our adulthood, especially if we can't learn to turn it off. So yeah, I think attacking it through the lens of inflammation has real promise. I would add only that I don't think, and I don't think anyone in the field thinks, that there is going to be a single silver bullet that allows us to completely arrest the disease. I think in some ways you were hinting at it, that what we're going to need is a multi-pronged approach that involves lifestyle changes, as well as interventions like anti-inflammatories. So yeah, good question. And I think you're right on there.
Dr. Herrup, you knock out amyloid and you see side effects in normal adults. So number one, what is the functional role of beta amyloid? Like, clearly it does something in the brain. Is it upregulated as an inflammatory response? So that's part one. And then part two, can you draw any parallels between the way that we talk about Alzheimer's and the way we talk about cancer? Cancer, Alzheimer's, aging, it's the accumulation of mutations, in the body. And so I think that people throw their hands up when we start talking that way. How do we talk about the accumulation of mutations in a coherent way where people just don't throw their hands up? The case study is, my Nana smoked until she was 95 and never got cancer. And then the 40-year-old marathon runner gets a brain tumor and dies. So people throw their hands up and they say, well, I'm just not going to worry about it. But how do we talk about this in a more intelligible way with the public?
Oh boy, what a good question. So it's actually, there are multiple parts to it. So let me try and tease some of them apart. The mutation part, the DNA damage part, I'm of the mind, and it, again, I would list it as a hypothesis. I can cite you chapter and verse for why I hold these ideas. But I do think that it's this loss of DNA integrity, as I call it, the accumulation of small mutations, that really drives the aging process as a whole. And young people do not get Alzheimer's disease. I mean, there is no juvenile onset form of Alzheimer's disease. It is a disease of late life. It is very uncommon before the age even of 65. So aging, the study of aging per se, is going to be an important part of the study of Alzheimer's. To me, if you're studying aging, you have to be studying unrepaired DNA damage as well. And so, yeah, and now I've lost the second part of your question. So could you repeat it just one more time?
Well, I've described it to peers that, you're born with a set of mutations, you inherit a set of mutations, maybe. And so you think of it as a poker hand, you might have 3 cards out of 5. We don't know. You don't know how many. You have no idea what you're born with, and you have the rest of your life to accumulate the other 2 cards to activate cancer or Alzheimer's. And so,
I like that analogy. Yeah, that was right. It was the analogy with cancer. So yeah, indeed, I think cancer, more directly than Alzheimer's has been shown to be a stray mutation in an oncogene leads to cells not being able to control their division, and that forms the tumor. It's hard to draw a direct analogy to the brain because brain cells, neurons at least, don't divide ever. So that it's hard to draw a direct analogy. But that idea of a poker hand where you still need to draw one more to get a win. In this case, the win is a disease, so it's a loss. I think that's a good one. And I'd just one last shot is what my focus on DNA integrity implies, and your idea of drawing that missing card. This has to be going on at the level of the individual cell. That's what happens in cancer. But I think the people in brain science have been very slow to adopt that idea, because what it suggests is that you can have a cell in the same brain region that is very old right next to a cell that is still functioning as though it were very young. And how we deal with that variability and repair the one without damaging the other is quite a challenge. So yeah, good question. Thanks.
Thank you. Dr. Herrup, great presentation. Thank you so much. I'm kind of new to all this, so still learning a lot. Can you tell us if there are any countries that have significantly higher or disproportionate levels or numbers of Alzheimer's case, if there's an environmental or geographical perspective to it? And then the second part is there one country in particular that seems to be doing better work on Alzheimer's research, or is this truly an international effort? Thank you.
Sure. Well, I would say Alzheimer's is absolutely an ecumenical equal opportunity destroyer. There are some variations from country to country, but most of them can be traced back to life expectancy. A colleague of mine used to joke that Alzheimer's represents a triumph of modern medicine because we now get old enough to get it. So yeah, that goes back to my answer to the previous question about the importance of aging. And so the answer really is no. There's no significant difference anywhere in the world with the onset of the disease. Is anybody doing a uniquely better job? No. I think we're all struggling. And I could spend a lot of time sort of musing about how most medicine develops empirically. You try something, and if it works, you do it again. But Alzheimer's really has not been around that long, not because it hasn't been around, but because we haven't been able to get old enough. So our medical science, and I don't care if you're talking about, traditional Chinese medicine, Western medicine, doesn't matter. Those medical communities have not had it to deal with long enough to develop effective therapies. So yeah, it's an equal opportunity killer.
I have a question. You were discussing, is this been looked at as an inherited neurological disease, like there are others that people have inherited?
Yeah. So again, two basic answers to the question. So certainly people have looked at the patterns of inheritance very carefully. There are rare forms of Alzheimer's that are entirely genetic, but they are very rare. They account for 1% or less than the entire prevalence of the disease. Beyond that, there are, a lot of attention has been paid to what are called risk factor genes. They don't give you Alzheimer's, but they increase your risk for it. And we've come up rather short. We have a long list of genes that will statistically impact your risk, but the impact is very, very small, with I think maybe one or two exceptions. So yes, your inheritance matters. If you have a first-degree relative with the disease, you are at slightly increased risk for the disease. But again, that increase is not dramatic or anything to, lie awake at night worrying about. It's there, but it's modest.
Doctor, I appreciate that you've given us today. I'm not a doctor, I'm not a scientist, as many of the people here. I happen to be an unfortunate lawyer, I guess, and I just look at logic and practicalities. My late wife died about 4 years ago, my sister 2 months ago. I try to look back and try to practically look at what was different in her lives, or both of their lives. And I'm wondering if any studies been made on food, what you eat. So many times disorders today are caused by what you eat, what's in your body. And I wondered if any study been made. My wife and I are very much interested in vegetables and fruits and try to stay away from greasy foods. And we kind of believe that has something to do with it. Can you comment on that? Whether there's any studies on what you eat and the possibility of getting dementia or treating it?
Yes.
Thank you.
Sure, I can definitely comment on it, and it's also a good question. And the answer is, not surprisingly, yes. The simple way to describe it is what's good for your heart is good for your brain. And what I mean by that in particular, with respect to diet, is that anything that your cardiologist recommends to keep your blood pressure down, watching your salt, and taking medicine when needed, that aggressive control of your blood pressure has shown to be very effective in reducing your risk of contracting Alzheimer's disease. Of course, what goes along with that is not just the food you eat, but your exercise. And that is also actually proven to be one of the more effective interventions. And it's frustrating in a way, especially to me as a bench scientist, that it's, low tech, but it's effective. And so in the end, that's all we're really trying to do. So yeah, keep your blood pressure down, and diet is a great way to do that.
My question is about early onset Alzheimer's. I had a sister-in-law who was diagnosed at 46 and died at 55. And I was wondering if the studies have shown that the disease starts the same way in someone that young, or I know with her, it started in her eyes where she lost all peripheral vision first, and then it moved on. And I was wondering if you might have any information on that.
Wow. I've actually never heard of it starting in the eye, but it is true that there are rare genetic forms of the disease that start, one of their sort of common characteristics, they tend to start much younger. We tend to lump them under the heading of early onset, which is almost always genetic. We are aware of 3 what I would call disease genes that actually are fully what geneticists call penetrant, which means if you get the gene, you get the disease. And there's a pretty strong consensus in the field that the pattern of the disease is similar. I think there's a bit of quibbling still as to whether it's identical. But for now, we tend to accept early onset as just a more rapid form of the disease. So it is similar, yes.
And I'm actually a rotoractor, so I hope this isn't a cliche question. I'm glad to have the opportunity to ask it. For someone that's studied inflammation as long as you have, what are your thoughts on CBD? Because I know that there's been a lot of mixed, essentially opinions and results really on CBD lately. I also know they've passed legislation to where in a couple of years, we'll be able to get the results of research that's ongoing now, as opposed to having to wait that full year for it. But essentially, CBD is known to have properties that will reduce inflammation. So that's why I just wondered kind of what your thoughts on that were.
I mean, I guess I should be honest and say I don't have any specific thoughts other than I think it, as speaking as a basic researcher, I think it's a great area to explore for two reasons. The first is that I think, as I've alluded to and you pointed out, I've been studying inflammation as one of the, root causes of the disease for many, many years, and believe strongly that it is due in part to that altered inflammatory chemistry. The other thing is that for a variety of reasons, some of them not very good ones, research into the cannabinoids as a whole, which CBD is a member, has not been as aggressive as it could have been. And I think what information is beginning to come out is that they can be, they can be as valuable a research target as serotonin reuptake inhibitors are for the serotonin receptors. So I look forward to a lot of good research in that area.
These have been some incredible questions and a great discussion today. And Dr. Herrup, we are so grateful for you taking the time today to share not only your personal experiences, but your expertise with us as well. And what an awesome way to kick off our new Lunch and Learn series. I don't know, though. Do you guys think we should change the name to Lunch a la Carte? I'm thinking that we should just put him in charge of our marketing and branding. I think that is an excellent idea. And we're going to see some changes in our notes coming forward. But good job, Dr. Herrup. You have a whole new career ahead of you. Who knew?
I don't think so, but thanks anyways.
I want to remind each of you that the next session of this will be on Wednesday, September the 14th. And we do have Dr. Ben Barr from UNC Pembroke, and we look forward to having him. And then again in October, we will have Dr. Alan Levy from Emory University, and that date will be forthcoming soon. Now I'm going to ask each of you to do me a favor. When the video comes out on our YouTube channel, please share it on your social media accounts, share it in your club or district newsletters, and invite someone to join you next month. Not just another Rotarian, but someone who is not a Rotarian as well, because it's equally important that we share the message of CART and the importance of the research that our researchers are doing with people outside of our Rotary families.